Okay, so welcome to this next video in which we are discussing habituation. Okay, so we are currently discussing the gill withdrawal reflex, and more importantly, the habituation of the gill withdrawal reflex, which occurs within this sea slug, Aplysia californica. Okay, right, so we have described the gill withdrawal reflex. Now what we want to look at is the neural circuitry that underlies the gill withdrawal reflex. Okay, so the neural circuitry is exceedingly easy, basically. Okay, so basically there is a sensory neuron which innervates the siphon. Okay, so the portion of the siphon that you are dropping water onto will have a sensory neuron that innervates it. This sensory neuron will take the information from the siphon. Okay, and I'll separate this out just to make it look simple. Okay, so here comes the sensory neuron, and it will go to something called the abdominal ganglion. Okay, so this is going to be called the abdominal ganglion. Okay, and this will be in this sort of position on the Aplysia californica. Okay, so here comes this sensory neuron into the abdominal ganglion. Okay, and basically a ganglion is a name for a collection of cell bodies. What will happen is the sensory neuron will synapse on the next neuron along, okay, and that next neuron along will be a motor neuron, okay, and specifically this motor neuron that it synapses on is called the L7 motor neuron, okay. And basically, what happens is the sensory neuron will activate the L7 motor neuron, and the L7 motor neuron will then go and innervate the gill muscle, okay, and will cause contraction. So that's now just colour in these bits. So here's the gill in green here, and here is the siphon in red here. Okay, right. So basically, it's a monosynaptic reflex. The gill withdrawal reflex has only a single synapse within it. The neural circuit only has a single synapse, so it's called a monosynaptic reflex. Okay, so the water being squirted onto the siphon activates the sensory neuron, which I'll call SN for sensory neuron, which then synapses onto the L7 motor neuron, which then innervates the gill muscle and causes contraction of the gill muscle. Okay, and that will then cover the siphon, and that's the gill withdrawal reflex. Okay, what we now want to ask is, how does habituation occur? What changes occur to this monosynaptic system that we have here that then leads to the reduction in the gill contraction that you get for the application of the water onto the siphon? Okay, so there are three possibilities for where this could change. The first possibility is that the sensory neuron no longer responds in the same way to the water, i.e. the sensory neuron will become desensitized effectively to the water, so it won't respond as rigorously to the water. Okay, so it could be a change on the level of the sensory neuron. And if the sensory neuron fired fewer action potentials, then it would stimulate the L7 motor neuron less, and hence the L7 motor neuron would cause less contraction of the gill muscle here, and therefore you would get a reduced gill withdrawal reflex. Okay, we can rule this out. The reason is, you can do electrophysiological analysis on the sensory neuron, and you can see how many action potentials are firing along that sensory neuron. And basically, if you do this for each of our trials, so remember, we are doing repeated experiments where we bring a pipette of water in, squirt it onto the siphon of this Aplysia californica, and then watch for the gill withdrawal reflex. So if you look from trial to trial, and you look at how many action potentials the sensory neuron is firing, you find that it doesn't change. So the action potential frequency stays constant in the sensory neurons, i.e. even though the gill withdrawal reflex is habituating, the number of action potentials that the sensory neuron is firing every time you squirt the water on is exactly the same. Action potential frequency unchanged, okay, in the sensory neuron. Okay, so we can rule out that it's something that is changing on the level of the sensory neuron. The second possibility that we have is that it's something on the level of the synapse, i.e. that the ability of the sensory neuron to activate the L7 motor neuron goes down, okay? So something on the level of the synapse. And we'll come back to that one. That's the right answer, okay? The third possibility is that the L7 motor neuron's ability to stimulate the muscle goes down. So it's a change on the level of the muscle. The muscle becomes desensitized to the L7 motor neuron stimulation, i.e. the L7 motor neuron continues trying to stimulate, but the muscle basically stops listening and produces a reduced contraction even though it's receiving the same stimulation. Okay, we can rule that one out as well, because if we stimulate the L7 motor neuron directly with electrodes and produce action potentials, we find that the contraction that produces in the gill muscle, okay, is exactly the same whether the Aplysia californica has undergone the experiment and therefore is now habituated to the water stimulus or not, basically. So, if you stimulate the L7 motor neuron of a fresh Aplysia californica which hasn't been habituated, you get a certain amount of contraction of the gill. You get the exact same level of contraction in a Aplysia californica which has been through this experiment and therefore is habituated when you stimulate the L7 motor neuron directly. So it's not a change on the way that the motor neuron interacts with the muscle. So it is indeed a change on the level of the synapse. Now, what actually changes on the level of the synapse? Well, basically what happens is the change is presynaptic. Okay, so in this monosynaptic reflex here, the change that occurs is actually on the level of the synapse, and it's on the sensory neuron, not on the postsynaptic neuron, not on the motor neuron. Because basically, you would think that there were two possibilities here. Either the presynaptic neuron can be releasing less neurotransmitter, okay, or the postsynaptic neuron can have reduced the number of receptors on its surface and therefore is ignoring the neurotransmitter even though you're releasing the same amount. Basically, it's the presynaptic one. The presynaptic neuron releases less neurotransmitter. So basically, we are releasing less neurotransmitter onto the L7 motor neuron. Okay, now the question is why? What actually causes the reduction in the release of neurotransmitter? Well, let's just revise how neurotransmitter is released from an axon terminal. Okay, so here's an axon terminal. Now, on the presynaptic membrane, you have docked these special vesicles which contain neurotransmitter. Okay, so these are vesicles full of neurotransmitter, and basically, vesicles full of neurotransmitter are called synaptic vesicles. So here's a synaptic vesicle, and basically these synaptic vesicles are docked to the presynaptic membrane, okay, which is the membrane facing onto the next cell along, okay, so this is the process of the L7 motor neuron. Okay, so this is a synaptic vesicle here. Okay, right. So, what happens to cause the release of the synaptic vesicles from the presynaptic nerve terminal is that you get the propagation of the action potential along the membrane, okay, so along the axon, into the axon terminal, and then what does it trigger? It triggers the activation of voltage-gated calcium channels. So when it gets to this active zone where we have synaptic vesicles docked at the presynaptic membrane, basically you have special voltage-gated calcium channels in this area. And basically these voltage-gated calcium channels are activated by depolarization of the electrical potential difference across the cell membrane. And when they open in response to the action potential, because the action potential, remember, has a depolarization phase, okay, they will allow calcium to move into the cytoplasm. And this calcium then causes the exocytosis of the vesicles, the synaptic vesicles, into the synaptic cleft. Okay, basically, there is something that happens on the level of the voltage-gated calcium channels. They become less effective, i.e., when an action potential arrives in the axon terminal, the actual amount of calcium that you get coming into the cytoplasm of the axon terminal for a single action potential will be reduced. Okay, something happens on the level of these voltage-gated calcium channels that causes a reduction in the amount of calcium that comes in. And if you bring in less calcium, then the amount of synaptic vesicles you will actually exocytose will also be greater, because the calcium triggers the exocytosis of the synaptic vesicles. So, basically the changes on the level of these voltage-gated calcium channels, and it is not known what the actual change that actually occurs on these voltage-gated calcium channels is, but something happens. Okay, so something happens to reduce the effectiveness of the voltage-gated calcium channels, so that when the action potential propagates into the axon terminal, the amount of calcium that actually causes to enter the cytoplasm is reduced, okay? And that then leads to reduced exocytosis of synaptic vesicles, hence you get a reduced amount of neurotransmitter released, and that then leads to reduced stimulation of the L7 motor neuron, okay? So gradually habituation occurs, and the sensory neuron that's being stimulated just as rigorously by the water stimulus is going to release less neurotransmitter onto the postsynaptic cell. Okay, and that is how habituation occurs in the gill withdrawal reflex within the species Aplysia californica.